He's an associate professor in the Department of Comparative Medicine at Yale School of Medicine. And before starting his independent research at Yale, he did a postdoc at Rockefeller University in the lab of Jeffrey Friedman, who many of you might know for the discovery of leptin and uncovering its function in obesity. But in the lab, Matt was kind of the odd man out because he wasn't really interested in leptin. Instead, his interest lied in determining the identity of cells that generate the new adipocytes in mammals. And his pioneering work that started in his postdoc identified these bona fide adipocyte precursor cells in vivo, establishing markers to prospectively identify the adipocyte precursor cells. And the work allowed his lab and many others to continue to characterize and define the contribution of various adipocyte lineage cells to the growth of adipose tissue during natural development and diet-induced obesity. Matt's lab has been quite well funded, receiving multiple R01s throughout his tenure at Yale, and his group has numerous publications in really high profile, high impact journals like Nature Cell Biology and Cell Stem Cell. I remember from when I was doing my postdoc at Yale, when Matt received a very prestigious Helmholtz Young Investigator in Diabetes Award for his work. It's something that identifies the rising stars in the field. I wish I had a picture that I'd be able to share, because in celebration, Matt bought and wore lederhosen, which is just a wonderful image. Unfortunately, I can't share such a beautiful image with all of you. I've known Matt for about—
You're going to have it when you started going down that road.
I've known him for about 10 years and really have always admired his passion for his work, and more specifically, how he mentors individuals. So he's always been highly engaged with the trainees, very hands-on in their training, and really strives to produce peers. To elevate them up and have them rise to that sort of a level, something that I try to emulate with my team. So it gives me great pleasure to welcome you, Matt, to this virtual stage for this month's cross-disciplinary seminar series that's focused on metabolic disease. So Matt, when you're ready, please feel free to begin your presentation.
All right, thank you, Brett. Thanks for the opportunity to be here, and thanks for that wonderful introduction. And can you see my slide? Are you seeing it? Everything's working well? Okay, good.
Yes, everything looks good.
So as Brett already said, my lab has really focused, historically on really the role or the mechanisms that regulate fat mass. And of course, well, what I want to do today is I'll get a little bit into some of the stories that the history that Brett sort of brought up a little bit, but mainly as sort of a window into how we started to take a little bit of a new direction in getting really interested in how the types of fats we eat actually impact both obesity and metabolic disease. So of course, the lab, we're originally been studying adipose tissue. And if you have too much adipose tissue, of course, you're defined as obese. And I think you all know, but these are sort of those background slides we give, that, obesity has now become a global epidemic. And, when I was in my postdoc initially, right, there was a lot of, there were papers out and people talking about at the time how, yes, obesity rates had gone up, but they were stabilized and they weren't going to keep going up. And I think now we can see that hasn't been the case. So, not only, in other countries, but the US as well, we still have obesity rising at this time. And of course, that comes at an enormous cost. And currently, or the projections are that this is costing over $200 billion a year in the United States in total, which is really interesting to consider when you look at the NIH budget is, $42 billion a year. So we're dwarfing the NIH budget in our costs from obesity alone. Now, the reason that obesity has such a burden on healthcare is that somehow, there we go, is that obesity is associated with many diseases, most notably really diabetes, heart disease, and cancer, right? And so it's actually obesity's relationship largely with these other diseases that is responsible for its sort of outlandish cost. Now, one thing we do know is that where we gain your fat matters in terms of your risk of developing these comorbidities with obesity. If you have visceral fat, more common in men, of course, it comes with a relatively high risk of developing diabetes and metabolic syndrome. But if you have more subcutaneous growth of fat, which is more common in women, that comes with a relatively lower risk of developing these disorders and diseases. Now, we know at the very most basic level, right, there's two factors that really contribute to adipose mass increase. That is that the adipocytes themselves, these mature cells, can just accumulate more lipid and become bigger via hypertrophy, or you can get more adipocytes. Now, because these mature cells are actually post-mitotic, the generation of new cells must come from the proliferation and differentiation of precursor cells. However, before I started my work in my postdoc, we didn't know what those cells were. And if you don't even know what the cells are, it becomes very difficult, of course, to do directed studies of the mechanisms that are controlling their differentiation in the obese state. So this is one thing. So we had known a lot actually in the past about sort of the nuts and bolts of adipogenesis and sort of these transcriptional factor cascade, et cetera, that have been characterized, including PPAR gamma, that lead the precursors to become mature adipocytes. But these studies really have come all from in vitro cell culture models, most notably the 323L1 cell model developed in Howard Green's lab. And of course, if you're studying these, studying this process in a dish, it really doesn't help you understand how the fat mass is regulated in vivo, or how we get differential accumulation of fat in different depots, in this case, between the sexes. So for me, the question was really about trying to develop tools. The goal was to develop tools to be able to look at the regulation of fat mass in vivo. And as Brett sort of mentioned, in my postdoc, I did that by taking a flow cytometry-based approach to isolate out different cell populations, and really then identified this relatively rare population of CD24 expressing cells in vivo that are capable of actually forming an entire fat pad when they're injected into the proper site in fatless mice in vivo. And so us and our lab and others now have really done a lot to verify that indeed this is the adipocyte precursor lineage in white adipose tissue in vivo. So we have the cells and we know we can study. We also now in my lab have developed a whole host of genetic tools that allow us to study the adipocyte lineage in vivo as well. Now, the most important sort of breakthrough that we had that allowed us to do this was really identifying this membrane tomato, membrane GFP mouse that was actually, and what this is it's a membrane targeted tomato protein. So the membrane itself is red. That's flock stopped in front of a membrane targeted GFP. And when CRE is activated, you actually get a removal of the red label and now expression of the green. So you just get a red to green membrane switch. And that's really important for studying adipocytes because they have virtually no cytoplasm. So most of these CRE reporters, the CRE reporters we use in lineage tracing studies are cytoplasmic. You can't really use those for adipose. I tried when I was a postdoc, and this tool really allowed us to really make great strides in the field, and now basically become a very common tool that's used in the field. And just an interesting side note, Mandar Muzumdar was a graduate student when he published and generated and published this mouse, and he is now actually a colleague of mine in the physiology department at Yale. So we now have sort of a very well-established understanding of exactly how some of these progenitors work, that these CD24 cell expressing cells are early progenitors in a rare population that proliferate and differentiate to produce committed pre-adipocytes that can be held in vivo before they actually transition to fill with lipid and become mature adipocytes. What we really wanted to understand now that we had we had identified the cells was really how do they respond in obesity, and how is their differentiation regulated in obesity? Because there had been a lot of controversy actually about how prevalent adipocyte hyperplasia was and how critical it was for fat mass gain in obesity. And to really study this, their most common model that we use is the high-fat diet feeding model of C57 black 6 mice. You put these mice on a high-fat diet, most commonly a 60% or 45% lard-based diet. They gain weight rapidly, and that weight gain is due to an increase in fat mass. So one of the first steps we wanted to do was take advantage of our MTMG reporter model and an adiponectin Cre-ER, so an inducible version of a promoter that's only expressed in mature adipocytes, to determine what the time window of hyperplasia might be in vivo. And for this, we just take these all red expressing adipocytes, pulse with tamoxifen. Only the mature adipocytes then become labeled with green. We then do a chase, and it's sort of a reverse experiment where what we're looking for is the reemergence of those unlabeled cells, because the adipocytes are post-mitotic and don't divide themselves. So when we did this, at least Jeffrey did this, she found that after 8 weeks of putting the animals on a high-fat diet, we actually saw that there was an increase in visceral fat adipogenesis, the formation of new cells, over the controls, but that was only in visceral fat, not in subcutaneous fat. And she also then determined the size of the new and the old adipocytes. There was really no difference whatsoever. The cells are the same size. So if there are more cells and they're the same size, they contribute to fat mass. And indeed, we see that there is correlation between the formation of new cells and the weight of the adipose depot itself. Now, we studied, all of these studies were done in male mice, and this actually sort of corresponds with what we know about obesity in men as well, that we tend to accumulate visceral fat as compared to subcutaneous fat. Now that we knew that we were looking in the first several weeks of high-fat diet feeding in terms of when adipogenesis is occurring, we wanted to know when are our precursor cells actually responding to the high-fat diet. So for this, we put the mice on a high-fat diet, then we pulsed with BRDU for 1 week periods and took samples and determined cell proliferation in different one week increments over the time course. What we found is, I mean, still to me, a really interesting and kind of a crazy result, actually, in that we saw robust proliferation of these cells in the first week of high-fat diet feeding in the visceral fat, but that went back down to baseline for the remainder of our time course. And as is mirrored in the actual adipogenesis findings, we did not see this in the subcutaneous fat at all. So we're really looking at this very transient, robust, and rapid response to feeding in terms of the proliferation of the cells. And the question was, are these proliferating cells actually contributing to adipocyte formation? For that, we did a slightly different assay, where what we did was we put the mice on high-fat diet and BRDU for the first week of feeding, of feeding the high-fat diet. And then we removed the BRDU and chased them out to an 8-week endpoint, and then determined the actual proliferation or the incorporation of BRDU into the adipocytes using immunohistochemistry. And for this, and the very important point is that we label the mature adipocyte membranes with caveolin 1, and then only score the adipocytes that are inside of that staining as the mature adipocyte nuclei. And when Elise quantified the BRDU incorporation in these cells, she found that yes, indeed, the visceral adipocyte nuclei had an increase in BRDU incorporation, indicating that those proliferating cells from the first week of high-fat diet feeding are actually contributing to mature adipocyte formation in vivo. Now, what I've told you about so far is that was all done in male mice and showed that it's a visceral specific response, but what about female mice? So for this, we basically did several of the same assays. And when we did when we did the proliferation time course, we found again that looking at visceral fat, we see this transient increase in precursor proliferation, which is similar to males, but different from males. We found that females have a broader proliferation pattern and that they also have increased proliferation in the subcutaneous depot as well. Now, do these proliferating cells, do we also see an increased or a sort of a broader pattern of actual adipocyte hyperplasia. The short answer of doing this assay is indeed yes, that we see an increase in adipocyte hyperplasia and adipocyte formation, both in the visceral and in the subcutaneous fat of females. So females actually have a broader, and indeed the same as in males, we now see correlation of adipogenesis to depot weight in both the visceral and the subcutaneous fat. So indeed, hyperplasia is actually playing a role in influencing the pattern or the distribution of fat between the sexes, and that there's a different actual contribution of hyperplasia in the subcutaneous fat between male and female mice. So now that we've characterized all the basics at the cellular level of this, we really want to know what were the molecular mechanisms that might be driving hyperplasia. And for this, we really keyed in on the PI3 kinase AKT pathways early in these experiments because it was known to be involved in nutrient signaling. And when we inhibited PI3 kinase with wortmannin, we actually completely ablate proliferation of the precursors in response to high-fat diet feeding. And we then have done a lot more studies looking at the role of AKTs in this process. And it turns out that actually only AKT1, and AKT3 were not involved at all. However, when we perform experiments in AKT2 knockouts, we see that actually they have very little or much reduced response in terms of precursor proliferation to high-fat diet feeding in that first week. And then when we then score the incorporation of BRDU into the adipocytes, we see that there really is no hyperplasia in the absence of AKT2. So we require AKT2 in the whole body knockout. We also then did these experiments in mice that have AKT2 knockout restricted to the adipocyte lineage in fat, and found that indeed it was AKT2 within the precursors themselves that is required for hyperplasia. So one thing that was of interest to us, we had these AKT2 null mice, it was very important to sort of characterize whether they had a defect in fat in the first place. And we simply looked at adipocyte precursor proliferation at many time points during development and saw no difference. We then scored adipocyte size at different times in development as well, and found again that there were no differences in either proliferation, establishment, adipocyte precursor number, or adipocyte size. And if you don't have a difference in number and size, you don't have a difference in fat mass at all. So this really led us to conclude that AKT2 is not actually involved at all in the formation of adipocytes during development, but that it has a specific role in obesity, which of course led us in this paper to conclude that there is an obesity-specific mechanism of adipogenesis. And that was relatively rapidly followed up with and confirmed from Phil Scherer's group as well, and has now actually been confirmed by several more publications since. And so one of the biggest things to us that we really wanted to understand was what is really controlling this obesity-specific mechanism of adipogenesis. Right, we've known that it involves PI3 kinase and AKT2 signaling that leads to proliferation differentiation. But really, this happens and was stimulated by just simply feeding the mice a high-fat diet. So what is the signal that is driving this process? Now, one thing that happens, there's two main things that we know that happen when you put mice initially on a high-fat diet. One is that they transiently overeat that diet when they first see it. And so what we did was we pair-fed mice fed a high-fat diet, so they had the same amount of calories as mice on the standard diet. And we found that it didn't matter if we reduced their calorie intake to the control mice, they still had displayed the proliferation. They also have a disruption of their circadian feeding rhythm of feeding. And we also did a lot of experiments to manipulate that and found no effect. It didn't matter how we fed the animals and when we allowed them to eat, we still saw this increase in proliferation. So it wasn't anything about how they were eating. We then wanted to know if it was about what they're eating. So all of the experiments that I've told you about so far were done with this lard-based 60% high-fat diet, this really standard diet used in the field that is made by Research Diets. But that's not the only sort of high-fat diet that is out there or that is used in studies. There's also a Sirwitt diet that is based on coconut oil, and then a milk fat, the Western diet, which is actually milk fat or butter-based diet. And so what we did, Chris Church did initially, was simply feed the mice these diets and do the 1-week proliferation assay. And what he found, of course, was very interesting in that lard, whether it was 45 or 60, which is what all the studies I've shown so far were done with, you see this robust proliferation of the precursors. However, if they ate a coconut oil-based diet, whether the carb source was cornstarch or sucrose, we saw no proliferation whatsoever, even though it was a 58% fat diet. So our initial thought was that it's still possible that we'd be getting actual adipocyte formation, but maybe in a scenario where we're getting depletion of the precursors because there's no proliferation. So we did then our genetic adipocyte adipogenesis assay using the adiponectin-free MTMG mice. And what we found was that indeed the coconut oil diet does not induce any hyperplasia in the visceral fat of male mice at all. So they look like the standard diet control in terms of their adipogenic rate. So the type of diet was actually, the type of fat that was in the diet was actually driving it. Now, the issue here, of course, is that these were just diets that aren't matched in any other way. So there's other differences in micronutrients, et cetera, between these diets than simply the actual, the fats themselves. So we really wanted to understand what the role of the composition of fat and whether it was the composition of fat that was impacting the adipocyte hyperplasia in the obesity models, in these high-fat obesity models. So what we did was we developed with research diets a panel of diets that are 45% in fat and differ only in the source of the fat. And you can see here from just sort of a overview of the lipidomic data that we have a broad range of the composition of fatty acids in these fat sources as well. So as a first pass, a postdoc in the lab simply took these diets and did the 1-week proliferation assay again in the male mice, where we would only see proliferation in visceral fat on the lard diet. And what she found was that indeed there is actually a very nice gradient from no induction of proliferation to substantially higher induction of proliferation than even the lard diets that we used in the original studies, where there is no difference in the subcutaneous differentiation across this diet panel. So since we have a really nice gradient of responses, it really shows us that yes, indeed, the type of fat that these mice are eating is actually dictating whether or not they're undergoing a hyperplastic response. And we actually have done the MTMG genetic tracing assays to confirm this, that it matches with this. And so, oh, excuse me. So what we did next was actually did lipidomics on, took the lipidomics on the diets and did correlation analysis between the proliferation response and the types of fats, different fats in the diet. And we found that only one dietary fat, dietary oleic acid, actually correlates, and it correlates incredibly strongly with the proliferation response. And here, just as an example, we see some of the other more abundant fatty acids in the diet, and that there's really no correlation whatsoever. So it's very interesting to us that this really, one of the most abundant fats in our diet, appears to be involved in driving hyperplasia. So Chris Church, a postdoc in the lab, then took another approach to see if this was a direct effect on the adipocyte precursors themselves, or if maybe it was having an effect somewhere else in the body that was indirectly affecting the adipocytes. So he isolated the adipocyte precursors using flow cytometry and then cultured them and then treated cells with a whole panel of different fatty acids and found that oleic acid was capable of inducing differentiation, increased lipid filling in the cells in culture, and so was palmitoleate C16-1. But this was done at 100 micromolar fatty acid level in these studies. 100 micromolar is well within the physiologic range of oleic acid, but is a hyperphysiologic for palmitoleiate in these assays. So we really do believe that oleic acid is actually really the only driver of this process in vivo. But this data really supports that oleic acid has a unique function and it is actually acting directly on the adipocyte precursor cells themselves. Now, I showed you in our original studies that we had determined that AKT2 was a critical player in the adipocyte hyperplasia in vivo. And Chris also then looked at the ability that what happens to AKT2 activation when we treat with oleic acid, and he sees that indeed we see that oleic acid is capable of stimulating AKT2 in the cell culture as well. So this links sort of those previous studies to the role of oleic acid directly through AKT2. So we now know what I've told you so far. Oleic acid is able to drive the differentiation and proliferation and differentiation of adipocyte precursors to generate more adipocytes and increased fat mass in vivo. All of the work I've told you about so far has been done in mice. However, we have done a little bit of work to show that indeed this process seems to, or that the human adipocyte precursors also seem to be responsive to oleic acid with increased adipogenesis. So it's likely that this process is also playing a role in humans as well. Now, I really just wanted to sort of go into this historical background more as a means of sort of showing you how we got interested in studying dietary fats. And now basically I'll transition over to talking about sort of a somewhat different aspect of his studies. But we haven't really given up on this at all. We've been studying this mechanism a lot more, and I'm not going to get into the details on this really at this time, but we do now know that oleic acid actually it's working through LXR alpha signaling to actually stimulate the differentiation. And there's been links between oleic acid and LXR have been made before, but never really that direct. We also know that's sort of separately that estrogen signaling through ER alpha is actually also required. And what we don't know is sort of how all of these factors are playing together yet, and that's kind of where we are right now. So the interesting thing about it is that estrogen production in males, in male fat itself, is actually required to drive to signal through ER alpha to allow hyperplasia to happen. So it's even estrogen in the males as well that's required for this process. But anyways, so one of the biggest things, and when I originally got into this field and started studying all of this in the first place, really the long-term goal in my mind always was to understand what the role of adipocyte hyperplasia was in metabolic disease. Was it particularly bad? If we're driving the formation of these adipocytes, are these new adipocytes playing some sort of a functional role, that leads to metabolic dysregulation? But so one of the things we've been doing a lot for a long time now, along with these shorter term assays I've showed you about hyperplasia, we've been putting these animals on all of these diets long term and doing a lot of metabolic assays on them, and glucose, liver function, atherosclerosis, etc. And what we have found so far, it still holds true, it's been holding true for a long time, is that we have no aspect of metabolic disease that correlates with hyperplasia. Right? So this sort of made me honestly a little bit less interested in putting so much effort into understanding all of these roles of hyperplasia and the mechanisms of hyperplasia. But it turns out that almost sort of unintentionally, in a way, from how we got into it, this diet panel has turned out to be an incredibly powerful tool for studying metabolic disease, even just on its own. So when we do long-term studies and put these mice on these different fat sources long-term, we see a very widespread in the amount of fat that they actually gain, with the palm oil mice gaining more than double the amount of fat of some of the mice on different fat sources. Now, we've also done a lot of work looking at glucose metabolism. If we look at fasted glucose, right, There's also a broad range of fasted glucose, just dependent on the type of fat that the mice are actually eating. And then if we look at glucose tolerance, just a real quick refresher, glucose tolerance is actually what you do is you starve the mice overnight, you then inject them with glucose, and then you track the blood glucose over 2 hours. And you see that in this case, just comparing the standard lard diet to the chow diet, that there's increase in the glucose excursion, an increase in peak glucose. And so they're less able to handle that glucose load. And we commonly take the area under the curve of these plots to display the glucose tolerance. This is what I'm showing here for all of these different diets. And once again, like the fasting glucose, you see again that there's a broad range of glucose tolerances on these diets as well. However, right, we know that obesity is associated with diabetes, which is glucose metabolism, basically defect. And interestingly, though, what we see in this data set is that none of these things actually go together. So the GTT doesn't correlate with the fat mass, fasting glucose doesn't correlate with the fat mass, and the GTT, so that increase in glucose excursion in response to a glucose load, doesn't correlate to fasting glucose at all. This is because we are actually taking the incremental GTT in these studies. I should mention, we are zeroing out the fasting glucose in our data sets. So that's interesting. So we did the same thing again. What we did, we get very interested in the glucose. Well, we did. I have no idea what's going on. Is everybody seeing that? All right. So we then did correlation analyses of the different types of fats in the diet, 2 different parameters, the metabolic parameters we're looking at. Absolutely nothing in the diet correlates to fasting glucose. However, only one dietary fat, this dietary linoleic acid, the polyunsaturated fat C18-2, actually correlates very nicely with the glucose intolerance. And linoleic acid is actually an essential fatty acid. You have to take it in your diet. You can't make it ourselves. And so it actually makes perfect sense that if it's correlating from the diet, it's actually correlating from the plasma, that the plasma and the diet levels of linoleic acid are actually tightly linked. It turns out that if we do a similar analysis in humans, where we compare the GTT to plasma linoleic acid, we actually see a relationship again between the GTT and plasma linoleic acid, implicating that what we're seeing here in mice may also actually be occurring in human, that linoleic acid is impacting the glucose tolerance in both mice and humans. So it's also, I think, important to note, right, that GTTs, these glucose tolerance tests, are actually something that we use experimentally a lot to study glucose metabolism, but they are not used actually as the increased glucose excursion in GTT is not something that is used to diagnose diabetes in humans. So that's why it's actually a little hard to actually find good data sets and studies that have been done where there's been a GTT. And this I got from samples from Lou Fontana from a study he did while he was at Washington University in St. Louis, and able to actually do this analysis. Because normally from humans, it's increased fasting glucose, or it's simply an increase in glucose at the 2-hour point post glucose load, or an A1C level that are used to diagnose diabetes. So it looks as though linoleic acid may actually be playing a role in glucose tolerance in both mice and humans. And it turned out, so next what we really wanted to do was try and do a similar study looking at, or a similar set of analyses, looking at the gold standard assay for insulin resistance, which is the hyperinsulinemic euglycemic clamp. So in this studies, what you do is you clamp, or you actually infuse in a high level of insulin at a constant rate, and then you infuse glucose in at whatever rate it takes to hit a target blood glucose level. So the less glucose that you have to infuse in, the more insulin resistant the animal is. In this case, you just see the saline and vehicle, the controls versus the intralipid, which is a common lipid is the only lipid really that's been used to induce lipid-induced insulin resistance in this case. And you see that there's a lower glucose infusion rate with intralipid. Now, it turns out that all of the studies using this gold standard technique from the past actually have high levels of linoleic acid. Why is that? Is because intralipid and the similar products that are approved by the FDA that have been used in all of these studies, both in mice and in humans, actually contain a high amount of linoleic acid. And that's by design, because these FDA-approved products actually exist to treat linoleic acid deficiency. But that's the only thing that have been used. So what we wanted to do was determine whether or not an oil with no linoleic acid had a similar impact on glucose infusion rate in a clamp. And for this, we made our own emulsions, both with basically a high linoleic acid oil and a low linoleic acid oil. And really, this high linoleic acid oil is simply, our attempt was to make it just like intralipid, and we actually were very close to that when you look at our own lipidomics of the two different tissues. And then the low linoleic acid oil, and these were prepared exactly the same way. What you see is the high linoleic acid oil, just like intralipid, actually results in a lowering in the glucose infusion rate, indicating insulin resistance. Whereas really surprisingly to us, the low linoleic acid oil, despite the fact that we're putting in just as much lipid, really the glucose infusion rate, it looks just like water. So there's really no insulin resistance occurring whatsoever if you don't have linoleic acid in the lipid that you're infusing. So there's a lot that you can learn from doing these glucose clamps. And this is simply looking at the glucose infusion rate in a different way in a column graph, and see that there actually is, from the low linoleic acid oil to the high linoleic acid oil, quite a difference in the glucose infusion rate. If you look then at what tissues may be causing that difference in the infusion rate, we see that either the visceral and subcutaneous fat, there's really no difference in the effects of the two oils on glucose uptake. Suppression of glucose production by the liver, which normally happens in response to insulin, is not actually is not different, again, between either the high or low linoleic acid oil. But skeletal muscle, looking specifically in the gastrocnemius muscle, looking at glucose uptake, we see that indeed this is where you see a difference, where the low linoleic acid oil really has no effect on glucose uptake, where the high linoleic acid oil is inhibiting that. So linoleic acid seems to be affecting the muscle specifically. So what we then did next was we took our samples that we had from the entire diet study, from 5 mice in each diet, and we sent the gastrocnemius muscle out to Anna Culkin at the Baker Heart Institute in Sydney. She does in-depth mass spec lipidomics. And she did this, and she identified 587 lipid species that were present in all of the diets. And here you can simply see, based on the PCA analysis, that indeed there is a clustering of each of the diets with each other. The diets are actually impacting the type of lipid that's coming out of the lipidomic analysis, the composition of the lipids, and that these diets are clustering relatively well together compared to the other diets as well. So there's a diet-induced impact on the total lipid profile of the gastroc muscle. Now, there's 2 real lipids that have generally been implicated in driving diabetes and driving glucose metabolism defects, as diacylglycerols and ceramides. And of course, we see here that there, again, if you look just at the diacylglycerols and the ceramides, the diets do have a fairly decent impact on the composition and changes in the types of ceramides and diacylglycerols that are present in the muscle. However, when we did a correlation analysis to glucose tolerance of all the different individual lipid species, all 587. We only had 17 lipids that actually significantly positively associated with the glucose tolerance. And there were no DAGs, no ceramides whatsoever. And surprisingly, really only one diacylglycerol. Now, this diacylglycerol species actually is one that contains linoleic acid at both sites, right? So diacylglycerol is 2 fatty acids on glycerol. And in this case, we only see if there's linoleic acid on both. Now, also really sort of shocking to us from doing this analysis was that 16 of the 17 lipids that positively associate either definitely contain linoleic acid or likely contain linoleic acid, depending on the number of carbons and unsaturated bonds that they display. So I think that that's a significant enrichment, like a ridiculously significant enrichment actually, in linoleic acid containing species correlating with the GTT. But we were most interested in this linoleic acid DAG because the role of diacylglycerols in insulin resistance has been well characterized over the decades. And importantly, we also, a previous study in humans from a few years ago actually found that this dilinoleic DAG, the LA DAG, in human, again, skeletal muscle actually also associates with obesity and diabetes as well. So this really became the main focus for us, right? So it's known really that diacylglycerol functions by impacting novel PKCs, and in skeletal muscle, that's believed to be PKC theta. And it's actually even because there has been some evidence in the past that polyunsaturated fats on diacylglycerols can actually drive PKC theta activity. It's been hypothesized in the past that by Scott Summers' group that linoleic acid may actually play a specific role, but nobody has ever actually shown this yet. And now we have data that's suggesting that this may actually be the case. So we're very then interested, of course, in whether or not this role of linoleic acid is actually through PKC theta or not. And so we generated skeletal muscle specific PKC theta knockout mice. And when we put these mice on on chow or a coconut-based high-fat diet, the low linoleic acid oil, what we find is that there's actually no effect on the GTT in either of these cases. However, when we put these knockouts on a high linoleic acid oil, in this case, soybean oil, we see that there's a significant reduction in total area under the curve of the GTT. But I think really importantly, what you see is there's a reduction in the maximal glucose excursion here. So the peak glucose levels are actually reduced quite substantially in this theta, in the theta knockout. So this really indicates that that linoleic acids is actually impacting glucose tolerance through muscle PKC theta. So if we know that linoleic acid is having this function through theta, we wanted to really understand sort of like what exactly is this dilinoleic DAG, how is it impacting PKC theta activity? The role and the way that the model for how diacylglycerols activate PKC theta is that they bind to these dual, there's 2 binding domains. They're tandem repeats in the PKC, in novel PKCs that then bind to the DAG, that the DAG binds to. And then what that does is it releases this inhibitory peptide from the active site and now allows PKC theta to phosphorylate its substrates, which include IRS1, et cetera, that actually inhibit insulin signaling directly. Now, people have done a lot of assays previously looking at PKC theta activity and really shown that there hasn't been much of a broad difference in sort of the effectiveness or the activity of the protein of PKC theta by manipulating the fatty acid chains in the diacylglycerol. But we wanted to do but nobody has ever looked at whether dilinoleic DAG had an effect. And when we did these assays, this is again Rocio Saavedra, we purified human PKC theta. We do a concentration titration of the different DAGs with PKC theta, and determined actually that the dilinoleic DAG actually has both an increase in efficacy, so it drives a much higher activity of the enzyme, as well as having a lower EC50, so it's more potent. And indeed, this is the case even when, because of the higher efficacy, this is really an underestimate of the difference in the activity between these different ligands. So this really shows us that this C18-2, this dilinoleic or LA DAG, is really driving a much greater PKC activity. And we think that this may be why how this is driving insulin resistance. Now, one thing that we're really curious about was all of the previous literature, everything that's been done, it's decades worth of literature, really points to only this SN2 position, fatty acid, being a dictator of the activity of the novel protein kinases, Cs. And so we were wondering whether this having linoleic acid at the SN1 position was really driving anything, or if we only needed linoleic acid at the SN2 position. So Rocio did this where we basically did each control, one that only had linoleic acid at SN2 and another one that only had linoleic acid at SN1. And it turned out that if you replace linoleic acid at either SN1 or SN2, you do not get that increase in activity, the same increase you see with the 18-2 DAG. So this is really something really novel in terms of just our understanding of how this diacylglycerol is even driving the activity of PKC. And from similar studies where you see this sort of increase in efficacy, the most likely mechanism is that this dilinoleic DAG actually stabilizes the active state compared to the other DAGs as well. So it's probably increased stabilization. So we're very interested in trying to determine exactly what the mechanisms of that are. And we're working with Karn Rainish here in cell biology on this right now as well. So I've told you that this dietary linoleic acid is actually driving this increase driving glucose intolerance in the mouse model. And there's evidence that it may actually be doing the same thing in humans as well. And this is incredibly important to understand how this may be happening and how different types of dietary fats and dietary fatty acids may be impacting metabolic disease, because there has been a profound change in the types of fats that we eat, especially in the last 70 years or so. Which of course corresponds completely with the onset of the obesity and diabetes epidemic. Now, in the United States, this is what this data is from, you can see that that soybean oil in pink here has become, the major fat source for our processed foods in this country. And of course, soybean oil is actually very highly enriched in linoleic acid, which is one of the reasons we've become so interested in it. And it's not just the fat source that has changed, right? We've also changed over time how we process our fats. So initially, when we started introducing fats into the plant fats into the diet, in order to make them palatable, they were fully hydrogenating those oils. And then, of course, in the 50s with Ansel Keys' work and indicating that saturated fats are bad for your heart, we started to move away from full saturation towards partial saturation, right? And this is just an estimate based on very little data that I made myself of maybe what our changes in dietary fatty acid composition might actually look like over time. But so that would have resulted in a very drastic change. However, then in the late 80s and early 90s, was when we determined that trans fats, which are actually a byproduct of partial hydrogenation, are actually bad for your heart too. We then started moving towards simply not doing any of those processing the food, but including antioxidants in the lipids to keep them from spoiling. And this has led to an even greater increase in linoleic acid in our diets, shown here in blue. So not just the sources, but the ways that we've been treating the diets or treating our fat sources over time has really led to drastic changes in the types of fats that are in our diets. And I think it's really important that we sort of understand that. Now, this change is actually supported by looking back over time at samples from the freezers, from people's freezers and studies, and looking at simply the percentage of linoleic acid in adipose tissue over time, that it's been going up. So it's clear from many different sources that linoleic acid has been going up along with the increase in diabetes rates. So another thing that was interesting, something that we wanted to know, was whether or not this linoleic acid induced increase in glucose intolerance was actually is a reversible process. So we simply did a dietary switch model where we allowed the animals to stay on either the high or low linoleic acid diets for 12 weeks, and then did a switch of high linoleic acids diets to low linoleic acid diet, and then did GTTs to determine. If anybody wants to stack us a question, just jump on in.
I have a question.
Yeah.
I wanted to know if you were doing anything to have some kind of influence on the fast food industry. I find that from the low to middle income communities, which such as mine, there's a McDonald's and Chick-fil-A on every corner. You know, I can go in one direction, there's 3 McDonald's, another direction, 3 McDonald's. There's some where McDonald's are right across the street from each other, where you can, you know, if you go in one way, you can make sure you get a McDonald's, and etc. So if these things are aimed at these communities, I don't know how you can control obesity. Looks like if those things were not as available now, when I go into the more upscale communities like Bethesda, and you don't see all of this. You almost have to have a knowledge of where these things are located to even go to them. So what are your thoughts on that, actually, or do you have any information on if they are trying to decrease the number of these facilities that, to me, are driving the obesity epidemic?
So I think that is an appreciated issue, right? Is that particularly the one of socioeconomics, where it's the worst food that's the cheapest, really, right? These highly processed, high fat foods, high fat, high sugar foods are the ones that we've made the cheapest. And they say they can't afford to make better food. And I think, me personally, I see that as kind of being ridiculous, because farm subsidies pay them, the government is paying the farmers to make the food that we then, and because of that, that is really the biggest reason why it's so cheap, right? So we are subsidizing the production of this food that is so unhealthy for us. And I think that really, I don't hear anybody really pushing too hard on that right now. I mean, some people are, but it's something that's not getting enough consideration, I should say. Okay, so this is the last actual data slide, just showing basically that, and this is just the weights up top, but most importantly, point out the GTTs on the bottom, that when they were on, of course, a high linoleic diet, for the first 12 weeks, you see a higher or they're more glucose intolerant than on the low linoleic acid diet. But that if you switch from a high linoleic diet to a low, you actually basically get rid of that increase in glucose tolerance. So this is a reversible process. So you can actually, by changing diet, actually impact this, even if you're, so even if this is something that's leading to diabetes in a given individual, it should be reversible, which is good news. So once again, just to sort of end up where we started a little bit, this idea, of course, that there are all of these obesity associated diseases at this time. We now actually, I sort of think about it a little bit differently in that dietary fatty acids and maybe the change in dietary fatty acids and also the increase in fat intake in our foods, because that's been happening too, are maybe a more of a central player. And actually, we're actually studying mechanisms because, of course, one thing I didn't get into at all is another aspect of our study showing that actually these animals fed these different diets choose to eat different amounts of the diets. So the reason that the palm oil fed mice gain so much weight is they're eating 30% more calories than some of the other diets. The animals fed some of the other diets. So that's the choice. And we've shown now that that's actually related to dietary palmitate. And I'll tell you the honest truth, we've been struggling finding the actual mechanisms for that, but it has a huge impact on their preference for the food. Once they are trained on a high palmitate diet, they have to be trained. They don't have a naive preference for it. Once they are trained on it, they will always prefer that high palmitate diet in the future, even if you don't show it to them for several more months afterwards. So it's pretty incredible. But we also have data. We're looking actively, not just at the diabetes and the obesity, but also at atherosclerosis and cancer, working with John Wasilmirsky. And really interesting, atherosclerosis, our data actually sort of reconciles some of the issues about dietary fats that are out there with that, in that there are actually two separate mechanisms driven by dietary fats. One is driven by palmitate, which fits with the original studies about saturated fats being bad for your heart. But also, a little more surprisingly and independently, we see dietary oleic acid also drives it. And it's by two separate mechanisms, the oleic acid and palmitate. And we're really interested in whether or what's going to happen, because palmitate and oleic acid are in our bodies all the time. We make them. So we're really interested in, if we target both of those pathways at the same time, what that impact is going to be on the development of atherosclerosis. So anyways, this is a lot. I got to say, one thing I've learned from, I guess I don't think I've actually learned it. I'm still learning and refusing to learn, maybe is the appropriate way to put it, that your lab can really get stretched too thin if you go in too many directions. So anyways, just real quick, the lab members who did a lot, Rocio Saavedra Pena did a lot of work with the PKC theta, both the knockouts and the enzyme assay. I mean, just really developing that enzyme assay was a huge task, and she got it done well. Jasper's done a lot of work with the emulsions, and Jen Kaplan was a previous postdoc who initiated the whole diet screen in the first place. With that, take questions. Thanks.
I just have a quick question.
Mark, go ahead.
Thank you. So firstly, just the previous speaker talking about the location with the lower socioeconomic areas, that's universal. That's the same here in New Zealand. So that's not your imagination. That's specifically by design. The observation I wanted to make, but I just want to check that I was right with that, was that effectively, just like in the past we were told incorrectly that a calorie is a calorie is a calorie, it appears now that we've also been told that a fat is not a fat is not a fat. And that depending on, and I'm also reflecting on linoleic acid with the health food industry, because of course linoleic acid converting to alpha linoleic acid, there are some supplements that contain these products that we're told are healthy, particularly in the vegan and vegetarian industry. Now it appears that actually we may need to reflect on that. Would that be fair?
I think that that's fair. I think all of these things we need to figure out, right? I think, you get to the sort of the socioeconomic aspect of it, right? Like there's another way of looking at the graph I showed with the oils. So that was per capita, right? But the population is growing. So if you actually look at it, not per capita, but you look at it in total production, really what you see is down at the bottom here, the red and the blue, those are the animal fats, right? There really hasn't been a lot of difference in production of animal fats over time, but we still need fat to feed the people. And what we've really been doing is layering on top of the animal fat that's available, all of these different plant sources. So I think really, to me, what it comes down to is figuring out a way where we can, what can we do to generate I guess you'd say, healthy fats, so healthier food to feed everybody. It becomes part of the issue, right? So, but we need to understand more. That's for sure.
I find it interesting that coconut oil has been vilified over the years. And yet, according to your research, actually, it's a really good oil to have in our diet.
So coconut oil is really unique, actually, because it's the only one that has really high levels of laurate, so C12. And that's short enough that it doesn't need the carnitine to be shuttled into, it doesn't need to be shuttled into the mitochondria to be burned. So it's more readily oxidized. So it's actually, honestly, I mean, there's one study I was working with Sue Keck where she was doing one cancer. She had preliminary data in one cancer model that maybe coconut oil was not great for that cancer. That's the only one that I would study that I'm aware of where the coconut oil was actually bad, to be honest. So,
well, I hope the American Heart Foundation's listening.
So we have a question in the chat from Serena Kajani. She says, great talk, thank you. And do you think there's a difference in terms of the response based on the source of linoleic acid? So the difference between linoleic acid from flaxseed and walnuts, which is higher, versus the soybean and canola oils?
I mean, I don't know because we haven't done anything, but I would imagine that if you're getting it from, same as with your sugar and fruit versus a soda, right? I would imagine that if maybe if it's not in this highly processed oil, which is probably taken up so much faster than it would be if you ate some nuts, that probably is going to, that if it's in the nuts and it's taken in slower, it would be less bad, I would think, right?
So I have a question with the type of diet, the type of fat that's in the high fat diet over time. So, and of course, I know there's a lot to do with this, and it's also expensive. But have you done anything where you keep or you get an animal obese on one high fat diet and then switch to another type of high fat diet, not remove the high fat, but maintain high fat for longer term? Because
so we did that here, right? This is one example of it. And this was,
so this goes from high to low, or just high linoleic acid to keeping high linoleic acid. So I'm wondering, because you have not only lipids that are going to be stored in the adipose tissue, but then you have a lot of lipids that are going to be in circulation. And I'm wondering, if you have a stockpile of a certain type of lipid in your adipocytes, if that is contributing to some of this, or if it's really about serum, what's in it, right? Like what's in your body right now that your cells are able to be bathed in and sense.
Right. There's a lot, I could go down a lot of different avenues on that. These are the types of questions I'm thinking about a lot, right? So one is like, a big one to me would be if linoleic acid is specific and this dilinoleic diet really is specific to muscle insulin resistance, why? Well, it probably, I would think, would have something to do with it being an essential fatty acid, that it's somehow being used as a readout of something, right? Some sort of energy status readout, right? I've really become interested in the idea that, all these lipases, there's many different lipases, right? So when you're mobilizing fat from, say, your adipocytes, right? Depending on how you do it, it's a specific lipase. And those lipases, although it's not really well known and not really well characterized, we know they have, just like you can hardly imagine that they wouldn't have a specificity for specific triglycerides, right? So that, and so that depending on what pathway you go down, you would be releasing different fatty acids, maybe different diacylglycerols, et cetera, based on which lipase you're using. That's one interesting thing to me. And then based on that, right? Like depending on maybe you're dieting or you're doing something else, are you retaining specific triglycerides in your fat, right? Versus releasing them. and how does that, those are all sort of interesting things to me as well. And sort of like start thinking about all of that. Like, how does this fit together?
Thanks. Ron, would you like to ask your question?
Yeah, so great talk. I really enjoyed it. I have two questions related regarding the sexual dimorphism. You described that early part of your talk. Number one, do you have any possible underlying mechanisms you could elaborate? Second is that technically, do you think that the tamoxifen treatment somehow could complicate the interpretation?
Well, so you mean that we use the Cree-ER for the tamoxifen?
That's right. Yeah.
Yeah. So that's a question that comes up a lot. So in our exact study, right, since we're doing the sort of the negative, right, it actually would lead to an underestimation of adipogenesis if tamoxifen was an issue. That's one part. The other part is we're actually careful to use a proper amount of tamoxifen, and we have I guess we're sort of lucky in that our Cree-ER actually worked very robustly. So yeah, it is, of course, you can, and that's the way that the Cree-ER was designed and mutated, was to respond better to tamoxifen than the non-mutated. That's why it's the T2 allele, than the non-mutated ER, right? So it's like if you put in too much tamoxifen, that's when you're going to hit the endogenous estrogen receptors, right? But it's actually made so that you don't have to do that. So if you dose it right, you should not have that problem. Right. And you had another question. Sorry. What was the female, the differences in the Adipose and about what the mechanism is. So yeah, we have actually a lot of data on that. I hinted at it a little bit. And some of it's been published, is that it is actually estrogen. So if we take we published a few years ago that if you treat the males with physiologic levels of estrogen, you actually will see a significant increase in subcutaneous fat proliferation, but you don't see nearly the level that you do in females. But we now know that actually the adipocyte precursors, one of the most highly expressed genes in adipocyte precursors, is actually in males only, not in females, only in the males, is estrogen sulfyltransferase, which deactivates estrogen. So that was really one of our first hints that maybe estrogen regulation is massively important even in male adipocytes. And so now that's something that Rocio's main thesis work has been on, is showing now that estrogen receptor alpha is required for hyperplasia in male mice too, and that the estrogen is actually coming from aromatase activity in the endothelial cells in fat. So it's a crosstalk between the endothelial cells and the precursors, and it's actually really, really, really cool. The real problem with all of those studies, and I think one of the reasons why probably not that many people really try to study sex hormones that much, is that as soon as you tweak one part of it, like we thought we had it, we knocked out estrogen receptor alpha using the PDGF receptor alpha Cree, and what we got was results that were, after we did more work, we showed that what happened was we blew apart all of the hormones, right? Estrogen levels went sky high, everything else, everything changed, testosterone was changed. And what we were looking at was really all of these differences. Those were the phenotypes. It had to do with all of those different levels. It had nothing to do with the ER alpha in the progenitors. So what we had to do for that was do transplants of the knockout cells into wild type animals to be able to determine what the requirement for ER alpha was. It's really hard to do sex hormone studies, we found. You just touch something somewhere and everything blows up. If you go back and look at some of the studies that have been done, you treat with aromatase inhibitors. Actually, what happens is in many of the studies that they've shown that the estrogen levels actually stay the same and all the other hormones go sky high in order to make sure that you're still producing estrogen. So you're not studying all these conclusions from a lot of those studies about the role of that phenotype is due to a lack of estrogen is not accurate. So it's another one of those things. It's like the Rocio now is like, I am never studying sex hormones again. So.
Great. Well, I don't see any other questions in the chat or any hands up from any of the participants still here. Another question just came through. What about fats in the liver? Did you look at liver in any of your experiments?
Yes. So we've done, actually, I'm really shocked. So we actually even did the in-depth lipidomics that I kind of showed a synopsis of in the liver as well, in the entire data set. And I really thought that, so of course the model, the idea is that when you have increased fasting glucose, that's due to a lack of insulin suppression of glucose production in the liver, right? So I thought sort of for sure, even though we don't see any dietary fats correlating to fasting glucose, I thought for sure we were going to see some lipids in the total lipidomics. And really remarkably, we see absolutely nothing correlate the fasting glucose in the liver lipidomics whatsoever, which blows my mind even more, given that the number of metabolites we're looking at, you would expect to have at least a couple just randomly, but we get nothing. So we have looked a little bit. We've been looking at that a little bit, and we just don't have much to say about the liver at this point, right?
Great. Well, thank you very much, Matt. This has been a very on point and insightful presentation for this seminar series. We appreciate your time and this talk, and everyone here, there will be another one next month, so keep a lookout for the advertisements that go around for the next cross-disciplinary seminar series. Thank you, Matt.
All right, thank you. Right, bye. Thank you, guys. Bye.